Later, we are going to continue like the mutator phenotype, mutant genes involved in DNA repair. If one considers cancer as a disease that results from alterations in the DNA of somatic cells, then it follows that any activity that increases the frequency of genetic mutations is likely to increase the risk of developing cancer. And nucleotides that become chemically altered or nucleotides that are incorporated incorrectly during replication are selectively removed from the DNA strand by DNA repair. DNA repair process requires the cooperative effectors or efforts of a substantial number of proteins, including proteins that recognize the lesion, remove a portion of the strand containing that lesion, and replace the missing segment with complementary nucleotides. If any of these proteins are defective, the affected cell can be expected to display an abnormally high mutation rate, which is described as a mutator phenotype. Cells with a mutator phenotype are likely to incur secondary mutations in both tumor suppressor genes and oncogenes, which increase their risk of becoming malignant. Then microRNAs, a new player in the genetics of cancer. MicroRNAs are tiny regulatory RNAs that negatively regulate the expression of target mRNAs. Given that cancers arise as a result of abnormal gene expression, it would not be surprising to discover that miRNAs are somehow involved in tumorigenesis. In 2002, it was reported that the locus that encodes two microRNAs, MIR15A and MIR16, was either deleted or underexpressed in most cases of chronic lymphocytic leukemia. It was subsequently shown that these two miRNAs act to inhibit the expression of the mRNA that encode the anti-apoptotic protein BCL2, a known proto-oncogene. In the absence of miRNA, the oncogenic BCL2 protein is overexpressed, which promotes development of leukemia. And because these miRNAs act to inhibit tumorigenesis, they can be thought of as tumor suppressors when the leukemia cells lacking MIR15A and MIR16, who are genetically engineered to re-express these RNAs, they underwent apoptosis as would be expected if a missing tumor suppressor activity was restored. The locus that encodes MIR15A and MIR16 is also deleted in other types of cancer, suggesting it has widespread importance in tumor suppression. The expression of two of the most important human oncogenes like RAS and MYC have also been known to be inherited by an mRNA, namely LET7, which was the first miRNA to be discovered. LET7. Some of the miRNAs act more like oncogenes than tumor suppressors. One specific cluster of miRNA gene, for example, is overexpressed during the formation of certain human lymphomas. Overexpression of these miRNAs can occur because the gene cluster encoding them is present in increased number or amplified form in the tumor cells, or it can occur because the gene cluster is exceedingly transcribed as the result of overly active transcription factors, including MYC. When mice was genetically engineered to overexpress these particular miRNAs, the animal developed lymphomas, as would be predicted if the genes encoding them were acting as oncogenes. And overexpression can be oncogenic when the miRNA target the mRNA of key tumor suppressor genes such as TP53. This happens to be the case in many neuroblastomas where the TP53 gene is not mutated, but it is not expressed at normal levels. The abnormal expression of miRNAs has also been implicated as a causal fact in tumor cell invasiveness and metastasis, which elevates the interest in these RNAs to an even higher level. It is not yet clear how important miRNAs are in the overall occurrence of human cancer, but a number of microarray studies that survey large number of these tiny regulatory RNAs suggest that most human cancers have a characteristic miRNA expression profile, just as they have a characteristic mRNA expression profile. Studies suggest that miRNA expression profiles may serve as sensitive and accurate biomarkers to identify the exact type of tumor a person is suffering from at the best avenue of treatment. The studies also suggest that mRNA may serve a potential target for anti-cancer therapeutics. It may be possible, for example, to treat patients with synthetic RNAs that act as miRNA sponges. Therapeutic RNAs of this class would process sequence that were complementary to oncogenic miRNAs and would act to bind and sequester such miRNAs by blocking their carcinogenic activity. Then the cancer genome. Next topic, the cancer genome. All cancers arise as the result of genetic alterations. As was evident from the previous discussion, the gene involved in tumorigenesis constitute a specific subset of genome whose products are involved in such activities as the progression of a cell through the cell cycle, adhesion to a cell to its neighbors, apoptosis, and repair of DNA damage. Taken together, more than 350 different genes have been identified as cancer genes. That is, genes that are thought to have some casual role in the development of at least one type of malignancy. Over the past few years, a concerted effort has been made to determine which of these genes are altered either by point mutation, translocation, deletion, or duplication in various types of tumors. This effort has been aided by recent advances in DNA sequences that allow researchers to determine the nucleotide sequence of specific regions of genome much more rapidly and inexpensively than ever before. It was hoped that most types of cancers would be characterized by alterations in a relatively small number of genes. It has been known for quite a long while, for example, that a large percentage of melanomas exhibit mutant BRAF genes, and a large percentage of colorectal cancers exhibit mutant APC genes. Similarly, the various types of leukemias are characterized by specific translocations, as exemplified by the BCR-ABL translocation in CML, and in all of these cases, these mutations occur at an early stage in tumor development and are thought to be critically important in turning the cell toward an eventual malignant state. However, results from the initial studies of cancer genomes suggest that the same type of tumors taken from different patients possess widely different combinations of aberrant genes. This observation presumably reflects the many different routes that individual tumors can take to escape the cell's normal anti-tumor protector systems. These findings are very important, where mutated genes identified in a large number of colorectal cancers are shown as peaks in two-dimensional multi-mutational landscape. The height of the various peaks reflects the frequency with which that particular gene is mutated in its particular type of cancer. It is evident from this type of display that a small number of genes are mutated in a large proportion of tumors. This can thought of as mountains in the landscape. For the most part, these are oncogenes that cancer researchers have been focusing on over the years. Each type of cancers has its own characteristic complement of frequently mutated genes. In the case of human colorectal cancers, the three most frequently mutated genes are APC, KRAS, and TP53 tend to become mutated at distinct stages in the progression of this type of cancer. Mutations in both copies of APC genes are found in over 60% of the smallest benign adenomas of the colon, suggesting that mutations in this gene often represent the first step in the formation of colon cancers. Then larger adenomas as well as cell masses in the early stages of malignancy tend to contain mutations in one of the RAS oncogenes, namely KRAS. In contrast, the TP53 gene tend to mutate or become epigenetically silenced only at later stages along the path when the tumor is clearly malignant. Cells displaying chromosome instability, which is reflected in increasingly abnormal karyotypes, only appear after mutations in KRAS. How a small number of genes tend to be mutated at a relatively high frequency in colorectal cancer. A surprisingly large number of genes are mutated at lower frequency, less than 5% of cases. These have been described as hills. These are approximately 15 different genes representing the hills in the mutational landscape of colorectal cancers. While we can presumably see that the genes that are mutated at high frequency are important factors in driving the cells into malignancy. Considerably debate is centered on the roles of the genes that are mutated at lower frequency. Many of the genes represented by the hills almost certainly have a causal role in determining properties of the malignant phenotype, even if they provide the tumor with only a small selective advantage. Mutations that cause or contribute to the malignant phenotype are described as drivers. Other genes that constitute a hill may simply represent passengers. That is, genes that tend to become mutated for some reason but have no effect on the phenotype of this cancer cell. It may be a daunting challenge to determine which of these genes are drivers and which are simply passengers. In addition to the genes of the mountains and hills, there is a large number of other genes that show up in the mutant state at very low frequency in a population of tumors. Mutations of this class can be seen as the small circles scattered over each landscape, and mutated genes of this class are presumed products of the overall genetic instability that characterize cancer cells. On average, an individual tumor carries approximately 80 different mutations. Close examination of these locations of the white circles in the two landscapes indicate that only a very small number of mutated genes in these cancers are shared by the two individuals. This, in a sense, each person suffers from his or her own distinct type of disease. Even within a given patient, metastatic lesions that have spread to different regions of the body can have remarkably different complements of mutated genes. These differences presumably reflect the different genetic changes that occur during the growth of each secondary tumor. It is evident from this and a large number of other studies on other types of cancers that the mutational landscape of the cancer genome is highly complex. However, a close analysis of the mutations that make up the mountains of hills suggests that most of this large number of genes encode proteins that participate as components of a relatively small number of pathways. In one study of persons with pancreatic cancer, which is one of the most deadly and most treatable types, recurrent mutations were found in more than 60 genes, but for the most part, these mutations affected a core set of 12 cellular pathways or processes. Most importantly, half of these pathways or processes were disrupted in 100% of the tumor samples. In a comparable study of glioblastoma, the most common form of brain cancer, the majority of tumors exhibited mutations that affect three major pathways like P53, PRB, and P13K. This, as discussed elsewhere in this chapter, cancer can be thought of not simply as a disease of aberrant genes, but as one of the aberrant cellular pathways. Mutations in any one of a number of genes disrupt the same pathway, and this have the same consequence for the cells. For example, the P13K pathway in breast cancer can be activated by mutations in the catalytic subunit of P13K, P13K3CA. Amplification of the growth factor receptor HER2 mutations in the PI3K effector PKB-AKT1 or loss of the PI3K antagonist P10. A few of the most prominent pathways that tend to be deregulated in many different types of cancers were indicated, and looking at a cancer as a pathway disease rather than a genetic disease generates more optimism among drug developers because it suggests that disrupting or restoring any one of the key steps in a single essential pathway may be sufficient to derail the malignant cells and lead to tumor regression. And before leaving the subject of cancer genetics, it should be noted that the view presented here, that cancer is a gradual multi-step progression of individual point mutations, is not universally shared. Some researchers argue that the mutation rate in humans is not high enough for cells to accumulate the mutations necessary to become fully malignant during an individual's lifetime. Instead, they have proposed that carcinogenesis is initiated by catastrophic events that lead to widespread genetic instability over a relatively small number of cell divisions. For example, mutations in a gene involved in DNA replication or DNA repair. As occurs in case of HNPCC, might rapidly spawn cells carrying widespread genetic abnormalities. According to another proposal, cells that have undergone an abnormal cell division and possess aberrant numbers of chromosomes are likely initiators of cancerous growths. The best way to decide among these possibilities is to analyze the state of the genome in cells at a very early stage in tumor formation. Unfortunately, for the interest of both researchers and cancer patients, it is virtually impossible to identify tumors when they are composed of a small number of cells. By the time the tumors are detected, the cells already exhibit a high degree of genetic derangement, making it difficult to determine whether these genetic alterations are a cause or an effect of tumor growth. Then, next topic, gene expression analysis. A technology for analyzing gene expression using DNA microarrays or DNA chips was very important. And briefly, a glass slide is prepared that can contain anywhere from a handful to thousands of spots of DNA, each spot containing the DNA corresponding to a single known gene. Any particular sense of gene, such as those thought to be involved in growth and division, or those thought to be involved in the development and differentiation of lymphocytes or some other cell types, can be included within the microarray. Once prepared, the microarray is incubated with fluorescently labeled cDNAs synthesized from the mRNA of a particular population of cells, such as those from a tumor mass that has been removed during surgery or from the cancerous blood cells of a patient with leukemia. The fluorescently labeled cDNAs hybridize to spots of complementary DNA immobilized on the slide, and subsequent analysis of the fluorescence pattern tells the investigators which mRNAs are present within the tumor cells and their relative abundance within the mRNA population. Then, studies with DNA microarrays have shown that gene expression profits can provide invaluable information about the properties of a tumor. It has been found, for example, that progression of a tumor is correlated with a change in expression of a particular gene. Certain cancers that appear to be similar by conventional criteria can be divided into subtypes that have different clinical features on the basis of their gene expression profiles. The gene expression profile of an individual patient's tumor can reveal how aggressive or how lethal the cancer is likely to be, and the gene expression profile of an individual patient's tumor can provide clues as to which type of therapeutic strategy will be most likely to induce tumor regression. We can look at a few of these issues in more detail, level of which 15 different genes are found to be transcribed in two different types of leukemia, like acute lymphoblastic leukemia or ALL and acute myeloid leukemia or AML. These genes used which are named on the right side represent those which transcription showed in the greatest difference between these two types of cancer cell blood cell cancers. Each column represents results from a single patient with ALL or AML. So that different columns allow one to compare the similarities in expression of a gene from one patient to the next. Gene expression levels are indicated from dark blue. These are related to some figures, so I need to skip this portion. The earlier a cancer is discovered, the more likely a person can be cured. It is one of the cardinal principles of cancer treatment. Yet a certain percentage of tumors will prove fatal, even if discovered and removed at an early stage. This is apparent, for example, that some early stage breast cancer already contains cells capable of seeding the formation of secondary tumors or metastases at distinct locations, where others do not. This difference is determined the prognosis of the patient. It was found in a landmark study by the team of Dutch researchers in 2002 that the prognosis of a given breast cancer is likely to be revealed in the level of expression of approximately 70 genes out of 1000 that were studied in DNA microarrays. This finding has had important clinical applications in guiding the treatment of breast cancer patients. Those patients with early stage tumors that display a poor prognosis profile on the basis of their gene expression pattern can be treated aggressively with chemotherapy therapy to maximize the chance that the formation of secondary tumors can be prevented. If gene expression data is not considered, these individuals are not likely to receive any type of chemotherapy because there is no indication using conventional criteria that their tumor has spread. Conversely, those patients whose tumor exhibit a good prognosis profile might be spread the more eliminating chemotherapeutic drugs, even if their tumor spread more advanced. In recent years, several companies have introduced laboratory tests like Mama Print and Oncotype DX to analyze gene expression profiles of individual breast cancers in an attempt to help guide the best course of treatment for these patients. Even though these prognostic tests have been in widespread use for several years, their validity is still being evaluated in large clinical trials. It is hoped that DNA expression profiles can soon be used to improve the diagnosis and optimize treatment of individual patients with all types of cancer. Now we are going to learn about a major topic related to cancer, like new strategies for combating cancer. This is a very important topic. It is painfully evident that conventional approaches to combating cancer, namely surgery, chemotherapy, and radiation, are not usually successful in curing a person of metastatic cancer, that is, one that has already spread from a primary tumor. Because they kill large numbers of normal cells along with the cancer cells, chemotherapy and radiation tend to have serious side effects in addition to having limited curative value for most advanced cancers. There has been hope for decades that these brute force strategies would be replaced by targeted therapies based on our newly formed insights into the molecular basis of malignancy. There are several ways that a therapy can be considered to be targeted. It can be targeted to attack only cancer cells, leaving normal cells unscathed. It can be targeted to a particular protein whose inactivation leaves the cancer cells unable to grow or survive, and or it can be targeted to the cancer cells of a particular patient based on their unique pattern of somatic mutations. Although the cure rate for most types of cancers has not improved significantly over the past 50 years, there is reason to believe that effective targeted therapies will become available to treat many of the common cancers in the foreseeable future. This optimism is largely based on the remarkable success that has been achieved with a small number of targeted treatments that will be discussed in the following pages. Even though the successes have been scattered among as much large number of failed prospective treatments, they demonstrate that the concept of targeted therapy is sound. In other words, the success can be considered a proof of principle. Just as important, they give both researchers and biotechnology companies the incentive to spread time and money to continue the pursuit of better cancer treatments. The anti-cancer strategies to be discussed in the following sections can be divided into three groups, like those that depend on antibodies or immune cells to attack tumor cells, or those that inhibit the activity of cancer promoting proteins, and those that prevent the growth of cancer blood vessels that nourish the tumor. Those that depend on antibodies or immune cells to attack tumor cells, then those that inhibit the activity of cancer promoting proteins, and then those that prevent the growth of blood vessels that nourish the tumor. First one is immunotherapy. Then inhibiting the activity of cancer promoting proteins. After that, inhibiting the formation of new blood vessels or angiogenesis formation. Then some experimental pathways like the discovery of oncogenes. These are the major topics we are going to learn, and after that we will be having a quick summary or synopsis of all these topics related to cancer, which will be done as a separate video. So let's start with learning about immunotherapy. We have all heard or read about persons with metastatic cancers who were told they had only months to live, yet they defy the prognosis and remain alive and cancer free years later. The best studied cases of such spontaneous remissions were recorded in the late 80s by a New York physician named William Coley. Coley's interest in the subject began in 1891 when he came across the hospital records of a patient with an inoperable tumor of the neck who had gone into remission after contracting a streptococcal infection beneath the skin. Coley located the patient and found him to be free of the cancer that had once threatened his life. Coley spent his remaining years trying to develop a bacterial extract that, when injected under the skin or into a tumor, would stimulate a patient's immune system to attack and destroy the malignancy. The work was not without its success, particularly against certain uncommon soft tissue sarcomas. Although the use of Coley's toxin, as it was later called, was never widely embraced, many studies have confirmed anecdotal observations that the body has the capability to destroy a tumor even after it has become well established. In recent years, two broad treatment strategies involving the immune system has been pursued, like passive immunotherapy and active immunotherapy. So passive immunotherapy is an approach that attempts to treat cancer patients by administering antibodies as therapeutic agents. These antibodies recognize and bind to specific proteins on the surface of tumor cells being targeted. Once bound, the antibody either kills the cells directly or orchestrates an attack on the cell carried out by other elements of the immune system. So, passive immunotherapy is an approach that attempts to treat cancer patients by administering antibodies as therapeutic agents. These antibodies recognize and bind to specific proteins on the surface of the tumor cells being targeted. Once bound, the antibody either kills the cells directly or orchestrates an attack on the cell carried out by other elements of the immune system. Then, the production of monoclonal antibodies capable of binding to particular target antigens was initially developed in the mid 1970s. During the next 20 years or so, attempts to use these proteins as therapeutic agents were foiled for a number of reasons. Most importantly, the antibodies were produced by mouse cells and encoded by mouse genes. As a result, the antibodies were recognized as foreign and cleared from the bloodstream before they had a chance to work. In subsequent efforts, investors were able to produce humanized antibodies, which are antibodies that are largely human proteins, except for a relatively small part that recognize the antigen, which remains mousely. On the past few years, researchers have been able to produce antibodies that have a completely human amino acid sequence. Herceptin is a humanized antibody that directed against a cell surface protein like HER2 that binds a growth factor that stimulates the proliferation of breast cancer cells. Herceptin is thought to inhibit activation of the receptor by the growth factor and stimulate receptor internalization. What is that drug? Herceptin antibody. Herceptin antibody. Approximately 25% of breast cancers are composed of cells that overexpress the HER2 gene, which cause the cells to be especially sensitive to growth factor stimulation. Up until the development of Herceptin, patients whose tumors overexpressed HER2 had a very poor prognosis. Their prognosis is not greatly improved. For example, one study of 3,000 women with early stage breast cancer reported that Herceptin reduced the chance of recurrence of the disease by about 50% in a 4-year period. And recent trials suggest that survival can be increased by combining Herceptin, that another monoclonal antibody like Omnitag, that interfere with HER2 dimerization. To date, the most effective humanized antibody is Rituxan, which was approved in 1997 for treatment of non-Hodgkin B cell lymphomas. Rituxan binds to cell surface protein CD20 that is present on the malignant B cells in the approximately 95% of the case of this disease. Binding of the antibody to the CD20 protein inhibits cell growth and induces the cells to undergo apoptosis. Introduction of this antibody together with chemotherapy has reversed the prospects of people with this particular cancer. Over the past few years, a number of fully human antibodies have been tested in clinical trials against various types of cancers. One of these, called Vectibix, which is directed against the EGF receptor, has been approved as a single agent treatment of EGFR expressing metastatic colon cancer. Because it is human protein, Vectibix remains in the circulation long enough to be administered once every other week.